All right, hello. I'm Dr. Ben Newman. I study coronaviruses and I'm going to try to answer your questions. All right, so, centered. There we go. That's more centered. Yeah. Um, this next question came directly to me via email, and I talked to the person who asked it and says, "Is it okay if I do a video on this?" Because I thought it was a good question. And so you won't find this in the Facebook or Instagram or whatever the heck we're on, however you ask questions. But yeah, let's try and answer it anyway. So it's a long question. I'll boil it down. The question comes from like an IT perspective. Say you were to find a shoe on the side of the freeway and the shoe had a USB drive in the bottom of that. You could plug it into your computer, but there might be downsides to that because this random USB would maybe have some code on it, and the code could be good or it could be malicious code. And it would be hard, just looking at the thing to tell which it is. It would actually be impossible, I believe. So the question is, when we are doing an mRNA vaccination, like the Moderna or like the Pfizer vaccination, we are essentially vaccinating with information, which is correct. You're giving the cell information to make a spike protein. So there's a little spike protein. And then once it's made the spike protein, that'll get chopped into little pieces and fed to a bunch of immune cells, immune system cells, in various ways that will lead to an immune response with all the bells and whistles. Yeah, just the best. So, how do you know that when you put in this piece of RNA, mRNA, like for one of those vaccines, that you're not also getting some kind of malicious code? How do you know that the code is clean, in other words? And it's a good question, and there are a couple ways that you can address this, or that it has been addressed. So a thing that people have done, but which is not done for the vaccine, is you can basically respell the code. So there are three little letters of RNA, and you read them in groups of three. So 1, 2, 3, 1, 2, 3. And the thing is, there's nothing in a piece of messenger RNA that tells you exactly where you have to start. You have to start at three particular letters, but those three letters are 3 out of the 4 letters, and they occur fairly often, like 1 out of every 64, places where you could have 3 letters. And so you will get potential start sites all up and down a piece of mRNA, and those can produce quite different proteins at the end. So how do you know when you put in a piece of RNA what you're going to get out? Well, a good way to do it, and a way that people have done this, is to just take the piece of RNA, put it into a cell, or put it into the machinery from a cell that turns RNA into protein, basically, and then look what you get out. And if you just get out the thing that you're expecting, then that means that that's what this makes. And if you get a little bit of, 5 different things, then you can say, whoa, yeah, one of these things is the one I want, and the other 4 we don't want, and let's respell the gene in order to get rid of all the all the other stuff that we don't want. Now, in respelling, you potentially run into the same problem again, because somewhere in there you're going to have those 3 letters that make the start, and you can potentially read it another set of different ways. So the other thing that we can do to look at this is to actually look at the virus itself. So we have lots of different whole genome sequences for the virus, and no, they're not beside each other. Let's stack them up. So you can stack them up so that they all have the first base, second base, third base aligned just like that. And you can see in looking at those where there are changes. And so let's say there are changes sprinkled all throughout the genome, which is exactly how it is right now. And then you can say, well, all right, so here we have what we call the spike gene. This is the thing we're trying to show the immune system. In the spike gene, maybe there's 50 other things that this could spell. They're all going to be smaller, and they're all going to be less likely for a variety of reasons we don't have to get into. But yeah, possible still, and things to rule out. And what you do is you look down the list and you say, okay, when there are changes, what does that do to all these little guys, all the little things that we weren't expecting? And what you would see, so if the spike gene is also coding for a little internal gene that maybe has some other effect, what you would see is as the virus changes, it would change in various ways, but it would not change so that this one would, the little one would go away. It would keep that one there because that would be important. It would be doing something for the virus. And if nothing is preserved as you start to change things, if none of these other little guys are particularly, always kept the same, then you can have a pretty good idea that probably there's nothing else in that code that is important to the virus. Nothing that makes enough of a difference that it would help or hurt the virus in some way. So yeah, so you got logic, and you can just do the experiment, express the thing, and see what proteins you get. Those are both valid ways to do it, and both of those have been done for the messenger RNAs in question. They've been examined, and it looks as though pretty much the only thing you can make with a spike messenger RNA is a spike protein. And that's good, but it's also good that people are thinking, like Philip was thinking, and asking these questions, because those are real good, smart science questions. And so there you go. I thought maybe that'll interest somebody, and I wanted to make a little video on it. Thanks very much. This has been Ask Dr. Ben.